Good morning. My name is Simone Sarasso, and I am assistant professor at the University of Milan, Italy. And the title of my talk is Consciousness and Complexity, an Empirical Perturbational Approach. Inferring conscious states from physical brain properties represents a major challenge with remarkable scientific, clinical, and ethical implications. That is particularly true given the pitfalls of the behavioral approach, which represents the gold standard in the assessment of consciousness, particularly when it comes to the difficult cases represented by disorder of consciousness patients, in which brain lesions may severely affect the processing, the correct processing of sensory input, as well as the production of appropriate motor outputs. Over the years, we observed many attempts in developing empirical measures for the assessment of conscious states, often leading to contradictory empirical results, as is the case for the late ERP components, also known as P3B. On the other hand, despite being initially promising, several other measures, such as brain metabolism, global EEG alpha power, and EEG delta power, showed a lack of sensitivity as well as specificity when applied to difficult cases represented by clinical populations as well as pharmacological manipulations. In this context, we recently observed a surge of studies providing evidence for the reliability of empirical measures based on the notion of brain complexity in assessing conscious states throughout a wide range of conditions, including sleep, meditation, drug-induced alteration of the state of consciousness, including general anesthesia, as well as neurological conditions such as epilepsy, coma, and in general disorders of consciousness. Also using a wide range of neurophysiological and neuroimaging tools. So these couple of hundred studies we reviewed recently spanned over a decade. They made use of wide range of conditions as well as tools in assessing brain complexity, and were independently proposed by researchers endorsing disparate frameworks. These measures were particularly reliable in detecting the presence and the absence of consciousness, so that they were recently endorsed and reviewed and recommended by both the American and the European Academy of Neurology in the guidelines for the management of disorder of consciousness. These measures not only showed remarkable reliability when applied in the clinical context, but despite assessing brain complexity in a plethora of different ways and different flavors, they can all be traced back to a notion, an early theoretical notion and principle, put forward more than 20 years ago by the seminal study by Tononi and Edelman in 1998, called Consciousness and Complexity, in which consciousness is associated with a specific form of complexity, so-called relevant complexity, which represents the coexistence of a high degree of functional differentiation and functional disintegration within a system. So the conjoint presence of functional differentiation and functional integration is the object of all these measures. Today I'm going to talk about a specific empirical measure of brain complexity we developed here in Milan, which is a form of perturbational approach to brain complexity, and can be traced back to this theoretical notion of relevant complexity as the conjoint presence of functional differentiation and functional integration in the brain. Specifically, we made use of two neurophysiological tools, transcranial magnetic stimulation on one end, coupled with high-density EEG, and we use TMS in order to probe directly and non-invasively the cortical surface of our brains, and high-density EEG to record the echo of this perturbation. This is particularly helpful when it comes to clinical settings in which patients are not particularly involved in any task, and the fact that we can access directly the cortical mantle allows for bypassing sensory pathways that are, as I said, often interrupted due to brain lesions. With this in mind, we applied this perturbational approach in several studies under different conditions in which consciousness was lost due to either physiological, pharmacological, or pathological conditions. Generally speaking, this slide summarizes the results of these studies, tracing back to 2005 up to recent days, and can be summarized as follows. Generally speaking, all the conditions associated with the presence of consciousness, be it wakefulness or dreaming or ketamine anesthesia associated with profound hallucinations, or even pathological states of brain lesion associated with the presence of consciousness, such as the locked-in syndrome, for example, are associated with, generally speaking, preserved spatio-temporal patterns, complex spatio-temporal patterns of brain activation following TMS. On the other end, all the cases associated with physiological, pharmacological, or pathological loss of consciousness were associated with the more stereotypical, either confined local or global stereotypical wave in which basically the spatio-temporal patterns collapsed in. More recently, we developed a more formal way of assessing brain complexity, in that we quantified and assessed the complexity, algorithmic complexity associated with these spatio-temporal patterns derived from TMS pulses. Specifically, TMS-EEG responses and the spatio-temporal patterns originating from identity EEG upon the application of a TMS pulse were first binarized, and then the spatio-temporal matrix associated to this binarization was compressed using a compression algorithm calculating the amount of information that is generated by causal interaction within the integrated system represented by our perturbed brain. We thus tested the so-called perturbational complexity index across the all the set of measures we performed over the years, namely conditions in which consciousness was either present or lost due to physiological, pharmacological, or pathological condition, by first calibrating the measure in conditions, known conditions, in which we could assess the presence versus the absence of consciousness based on behavioral reports. The results you see here highlight the fact that in all conditions in which consciousness was preserved based on immediate, in red, or delayed positive reports, were associated with high value of perturbational complexity index. Vice versa, all the conditions in which consciousness was testified to be absent based on delayed reports, such as REM sleep or anesthesia, were associated with low value of PCI and therefore brain complexity. Based on the separation between these two categories, positive versus negatives, we could identify an empirical cutoff, an empirical threshold, based on which we could then, using this empirical threshold, slice through clinical population, clinical cases in which consciousness was affected, basically the disorder represented by the disorder consciousness patients. First, by using this empirical threshold called PCI star, we assessed the sensitivity of the measure on 38 minimally conscious state patients, so patients affected by disorder of consciousness in which consciousness was present at a minimal level, so was behaviorally present based on the fact that the subject could, for example, perform very simple tasks such as following a person into a room or following his own image on a mirror. So very subtle signs of behavioral presence of consciousness. In this case, PCI attained unprecedented sensitivity of roughly 95%, so positively identified 36 out of 38 MCS patients. Then we went to the unknown, represented by the so-called unresponsive wakefulness syndrome patients, or old notion of vegetative state patients. And PCI identified two main classes of UWS patients, namely low PCI values, and a set of patients which are clinically, based on behavioral assessment, judged as unconscious, which attained PCI values as high as MCS patients or as healthy control subjects. So we had to classify this patient as possibly as covert conscious UWS patients. So overall, PCI performed pretty well in also in clinical cases, identifying also possible covert signs of consciousness. But rather than only being applied as a reliable marker of consciousness, PCI and TMS-EEG in general, so the application of this perturbational complexity, may offer some powerful insights on the mechanism behind loss and recovery of consciousness. So let's start from this observation, the one I showed you before. Now I added the complexity being high or low in order to underline effects. Low complexity responses, being it physiological, pharmacological, or pathological, low complexity states, are invariably associated to the presence of a slow wave, very similar to those obtained during sleep, even in pathological cases. Now, we know a lot of the cellular mechanisms behind these slow waves in physiology. We know that they subtend cortical downstates, which are a very small fraction of seconds in which neurons fall into a silent state, hyperpolarized state. And we know that we can detect signs of this neuronal silence from outside, so from LFPs or EEG, for example, in the modulation of high-frequency activity. Interestingly, TMS-EEG associated slow waves in sleep and anesthesia are characterized by the presence of a slow wave associated with high-frequency suppression, here in blue. Not only during sleep and anesthesia, but also during pathological states represented by disordered consciousness patients. So low complexity disordered consciousness states, so due to brain lesions, the TMS-EEG response was a slow wave associated with high-frequency suppression, so possibly linking a physiological mechanism to the pathological loss of consciousness. Interestingly, it doesn't seem to be an epiphenomenon, so the occurrence of a local of a sleep-like downstate in pathological condition doesn't seem to be an epiphenomenon because it seems to be tightly linked to the ceasing of the development of PCI in these patients. So the timing of the occurrence of the downstates, represented by high-frequency suppression, was linked to the plateau of PCI development over time. So low PCI value attained so low due to the presence of a downstate. Even more causally, we saw that during the clinical evolution of disordered consciousness patients, the occurrence of a slow wave tended to be replaced by a more complex wave, disappearance of high-frequency suppression, so disappearance of cortical downstates, and a progressive recovery of brain complexity as measured by PCI. Now, all in all, this may point to the fact that while anatomical lesions and disconnections cannot be reversed, it may still be possible to reduce the impact of functional disconnection by manipulating intrinsic neural properties, so offering potential for future development of treatments in disorder of consciousness. With this, I'm done, and I thank you very much for your kind attention.
Welcome everyone to my talk about neural correlates of unconsciousness, complementary roles of oscillatory and aperiodic brain activity. My name is Janna Lendner, and I'm currently a resident in anesthesiology at the University Medical Center Tübingen, Germany. When we look at electrophysiological brain recordings, salient rhythmic activity, so-called oscillations, stand out, here in blue. In between these rhythmic bursts is aperiodic, noisy, broadband activity, here in red. When we use Fourier transformation to look at the power spectrum, oscillations occur as prominent peaks that rise above the exponential decline, the 1/f, of the broadband power. Many names have been given to both phenomena. Aperiodic activity is often referred to as broadband or 1/f activity or electrophysiological noise. As it does not have a fixed temporal scale, it is also termed scale-free activity. One way to measure this activity is by fitting a line to the power spectrum in log-log space, here in green, where the steepness of the decline is also called the PSD or spectral slope, here in yellow. Because of their salience, oscillations have been studied in a manifold of cognitive tasks. For example, in deep non-REM sleep, cardinal sleep oscillations such as hippocampal ripples, thalamocortical spindles, and neocortical slow oscillations occur coupled to each other via cross-frequency coupling, mostly in the form of phase amplitude coupling, where the peak amplitude of the spindle, in black, is nested in a certain phase of the slow oscillations, in red. The strength of the slow wave spindle coupling is relevant for behavior and correlates with overnight memory performance. Compare the EEG recordings in deep non-REM sleep to the ones in REM sleep and wakefulness. While the first one shows prominent waxing and waning patterns in the form of slow oscillations, REM sleep is not characterized by any oscillations. In fact, it looks close to wakefulness, although the participant is fast asleep. Additional monitoring of muscle activity, the so-called EMG, or the eye movements, the so-called EOG, are needed to distinguish REM sleep and wakefulness. To characterize non-oscillatory states, we might need to quantify non-oscillatory activity. And indeed, aperiodic activity distinguishes both non-REM sleep, in blue, and REM sleep, in green, from wakefulness, in magenta. You see here that the power spectrum drops off steeper during non-REM and REM sleep compared to wakefulness. Moreover, the slope is also different between non-REM and REM sleep, helping us to distinguish between these two sleep stages. Interestingly, aperiodic activity is especially modulated in nodes that are also relevant for overnight memory consolidation, such as the medial prefrontal cortex and the medial temporal lobe. Under the general anesthetic propofol, here in blue, which increases GABAergic inhibition in the brain, the spectral slope of the power spectrum becomes steeper than during wakefulness, here in red. Although aperiodic activity is vastly different, we don't see any of the network-specific changes we observe during sleep. Instead, anesthesia caused a global brain-wide change in aperiodic activity. Other states of reduced consciousness, such as tonic-clonic seizures in epilepsy, in red, and metabolic as well as hypoxic coma, in green, are also characterized by changes in aperiodic activity compared to wakefulness, here in blue. As you see here, both seizures and coma are also missing prominent oscillations. So, are oscillations and aperiodic activity two separate phenomena? The answer is probably no. Aperiodic activity is interleaved with oscillations. Here, an example of a cortical slow wave in sleep, in red, where the aperiodic activity, in blue, changes over the course of the slow wave, and it's vastly different between the oscillatory trough and peak. Interestingly, we found a mirroring pattern between spindle oscillations in red and mutual information in black, where information cycles antiphasic to the oscillation, coinciding with aperiodic activity. These observations led to our current framework of interleaved oscillatory and aperiodic network activity in sleep, both playing important roles in overnight memory consolidation. Oscillatory activity can occur either coupled or uncoupled, both in red. Coupled oscillations open up time communication channels between brain regions, whereas uncoupled oscillations are thought to facilitate local plasticity. Both synchronized states, coupled and uncoupled, have in common that their entropy and thereby information content is low. Aperiodic activity, on the other hand, is by definition not characterized by synchronization and therefore offers high entropy, enabling plenty possibilities for processing, transformation, and integration of information. Recently, we started exploring how information capacity and criticality change with levels of consciousness. Current theories assume that waking brain states operate close to criticality, thereby enabling flexibility and maximum information capacity. Digressions from this optimum should lead to unconsciousness. In line with this hypothesis, we found that states of unconsciousness, such as anesthesia with propofol in brown and seizures in pink, are low in information content but differ in the level of stability. LSD in green, on the other hand, a drug that results in lucid hallucinations, was characterized by a higher information content closer to the edge of criticality compared to normal waking in black. What is the underlying neurophysiological basis of aperiodic brain activity? As we have already seen earlier in this talk, coma, anesthesia, sleep, and epilepsy are all characterized by changes of aperiodic activity. Moreover, they all demonstrate a shift of the neural balance between excitation and inhibition, where both extremes result in dysfunctions of the system. A prominent example of increased inhibition would be general anesthesia, where drugs actively enhance GABAergic inhibitory interneurons. Unbalanced excitation that is no longer balanced by inhibition would lead to a buildup of electrical activity and a sudden discharge, such as seen in a generalized seizure, which is also accompanied by unconsciousness. One explanation could be that aperiodic activity is indexing this balance, and some recent evidence backs up this hypothesis. Electrophysiologic recordings showed that the 1/f fit indeed changed with the power of broadband activity, which has been identified as a proxy for population activity. Moreover, computational modeling showed that an increase of the inhibitory drive resulted in a reduction of aperiodic activity, leading to a steeper drop-off of the power spectrum. Compare orange and green. In addition, we observed the same effect in our recent recordings under anesthesia and during sleep. Interestingly, the drop-off was even steeper in REM sleep compared to non-REM sleep, in line with a higher rate of inhibition. Although maybe counterintuitive at first, optogenetic imaging in mice revealed that cortical activity is indeed reduced even further in REM sleep compared to non-REM sleep, and that this shift is accompanied by an increase of active inhibition mediated by parvalbumin-positive interneurons. Taken together, Aperiodic activity distinguishes wakefulness from different states of consciousness, including drugs, anesthesia, seizures, coma, and sleep. Aperiodic and oscillatory activity occur interleaved and may serve distinct functions. Aperiodic brain activity is high in entropy and thereby information content. It may be an index of the delicate balance between excitatory and inhibitory cortical activity. Thank you for your attention.
Hello, and welcome to this talk investigating the spatio-temporal structure and complexity of auditory responses in coma. My name is Athina Tzovara, and I work at the University of Bern in Switzerland. To start with a short introduction, in our daily lives, we are constantly immersed in stimuli of the environment, like sounds or images. These stimuli very often do not occur randomly, but have repetitive rules and patterns. Our brains are particularly efficient in learning these patterns and detecting outliers in anticipated events. Notably, our brains can detect patterns and outliers not only when paying attention to the environmental stimuli, but also in cases when consciousness is diminished. Here are examples from the literature on 3 different states of reduced consciousness, where detection of deviant stimuli is preserved. The first is a study in sleep where rodents can detect standard and deviant stimuli in wakefulness, non-REM, and REM sleep. The second study used intracranial EEG and showed that in wakefulness there are distributed frontotemporal brain regions that are sensitive to deviant stimuli. As consciousness fades away in sedation and anesthesia, this detection is restricted in auditory areas, but it is still present. Third, we have an example of a study in pathological loss of consciousness in coma. Here we can see that in awake conditions we have a clear MMN, which stands for mismatch negativity response, which represents a marker of deviance detection measured with scalp EEG. What is very interesting is that the MMN response is preserved in a subset of coma patients, and this is generally associated with awakening and good prognosis from coma. In our case, we aimed at examining the neural correlates of auditory processing and deviance detection in pathological loss of consciousness, represented by coma. In our studies, we focused in the very acute coma phase in the first 2 days after coma onset. We examined post-anoxic comatose patients who are treated with therapeutic hypothermia, which downregulates brain metabolism and has been shown to improve the patient's outcome. After a cardiac arrest, patients are sedated and their body temperature is lowered to 33 or in some cases to 36 degrees Celsius for 24 hours. After that, patients are rewarmed to normal temperature and hopefully regain consciousness and recover. In such a context, where a treatment is applied in the first few hours after coma onset, it is important to have accurate and early predictors of patients' outcome, in order to better understand the neural functions that are still preserved in the comatose brain. In our protocol, we presented patients with sequences of standard and deviant sounds and recorded their EEG signals. We did this twice in hypothermia, so the first 24 hours, and after rewarming to normal temperature, generally within 36 hours after coma onset. For each patient, we used a multivariate decoding algorithm to classify whether a given EEG response was a response to standard or a deviant sound. Here is an example of the EEG topographies and latencies that were identified by the decoding algorithm for one patient in the first and second day of coma, which are quite different one from the other. In the first day for this patient, we have one discriminant period at late latencies, while in the second day we have additional periods at earlier latencies. This was just an example of one patient, and now if we look at these decoding results at a group of patients and healthy awake controls, what we observed is that on average, decoding performance was quite high, not only for healthy controls, but also for survivors and non-survivors. Next, we evaluated how the decoding performance changes from the first to the second day of coma. An improvement in decoding performance was predominantly observed for survivors, suggesting that this can be a marker for the return of consciousness. Overall, we observed that first, the neural mechanisms supporting deviance detection can be preserved in the absence of consciousness in acute coma, and second, that they change in a way that when the neural responses to standard versus deviant stimuli become more distinct in time, so auditory discrimination improves, the patient is likely to awaken from the coma. Now, the next question that we asked is what exactly is different in neural responses of survivors versus non-survivors from an electrophysiological point of view. Here we can see EEG responses to auditory stimuli for an exemplar survivor, non-survivor, and a healthy control. As you can see, these responses are quite different even visually and have a high variability across participants. Our next goal is to better characterize the neural dynamics that support auditory processing in coma. To answer this question, the field of consciousness research suggests that there are two key properties of neural signals that support conscious processing. The first is the synchrony of responses, which is typically disrupted and diminished in the absence of consciousness. The second is the temporal structure of responses, which can be quantified via measures of complexity or via the steepness of the 1/f EEG power exponent, and it is generally lower in sleep or anesthesia compared to wakefulness. Although these two properties have been well investigated in physiological and pharmacological loss of consciousness, like sleep and anesthesia, it is less well known how they manifest in pathological loss of consciousness. Here, we made the hypothesis that neural synchrony and complexity in coma do not only reflect the presence or absence of consciousness, but also the patient's clinical state and may be relevant for predicting the patient's outcome. To test this hypothesis, we focused on the first day of coma, where patients were comatose, sedated, and hypothermic. So conditions where we can be quite confident that conscious access to the environment can be excluded, both for survivors and non-survivors. We started with a pilot group of 38 patients where we performed exploratory analysis. We assessed neural synchrony of EEG responses through the phase locking value, or PLV, which quantifies how consistent are phase differences in pairs of EEG electrodes. What we observed is that in the majority of electrode pairs, on average, the PLV was significantly higher for survivors compared to non-survivors. Here you can see a subset of electrode pairs with higher PLV for survivors in red and lower PLV in blue. Then we focused on these electrode pairs that were identified as different at the group level and computed the mean PLV per patient in the pilot group. Here we could identify a threshold based on the mean phase locking during the first day of coma that separates patients who later survive from those who do not. This finding is not very surprising because the mean PLV was computed on electrodes that were different between the two patient groups. So then we moved to a second group of patients, a validation group, which was not assessed for identifying neither electrode pairs nor the threshold in phase locking. In the second group, we validated and confirmed that the PLV in the first day of coma is an accurate and significantly above chance predictor of outcome. This gave an accuracy of predicting awakening and overall outcome at 87%, which is at comparable levels with other outcome prediction measures currently used in a clinical environment. Additionally, the PLV has the advantage that it is a quantitative measure that can be computed already on the first day of coma. Interestingly, the phase locking for every healthy control was above the threshold, similar to patients who later survived the coma. These results suggest that overall neural synchrony is high even when conscious processing is present in the case of controls, or when conscious processing will eventually return in the case of survivors. Next, we evaluated the complexity of EEG responses in coma and wakefulness. What we mean by complexity is that if we have a signal that is perfectly periodic and fully repeats itself as plotted in red, the signal will have low complexity. Now, if we go to the other extreme, where we have little or no periodicity in a highly stochastic signal, complexity will be high. Last, we have the in-between state, represented in blue, where we have some periodic components intermixed with some randomness or noise. Going back to EEG, we can compute the complexity as Lebesgue complexity, which quantifies the number of unique patterns in an EEG signal. First, in the group of healthy controls, we observe that the complexity values of auditory EEG responses cluster closely, and this is reflected in what are quite typical event-related potentials in response to auditory stimuli that can be seen on the right. Second, we move to coma patients, and in particular survivors. Overall, survivors had significantly lower complexity values than healthy controls. At the level of auditory EEG responses, this manifests as smooth and long-lasting responses, which are often observed in coma and hypothermia. Third, in the group of non-survivors, we observed quite a different image. First, there was no significant difference in LZ complexity of non-survivors versus survivors, neither of non-survivors versus controls. Second, the LZ complexity values of non-survivors showed a bimodal distribution, having either higher or lower values than healthy controls. This finding may seem surprising at first if we consider the literature in sleep or anesthesia, which suggests that neural complexity drops when consciousness fades away. However, one important point here is that we are looking at a very particular case of unconscious processing, which is a very acute phase of coma, where the integrity of neural circuits is rapidly changing, and in particular, in the case of non-survivors, rapidly deteriorates. Additionally, we also computed the 1/f spectral exponent of EEG power. Previous studies have shown that the steepness of the 1/f exponent is a proxy to neuronal noise and to the excitation to inhibition balance. So that suggests that if a neuronal population is highly correlated, this will result in steeper 1/f, while if there is more asynchronous firing, the 1/f will be flatter. Previous studies have shown that the 1/f slope becomes steeper when consciousness fades away, tested in sleep and anesthesia. Here in our data, the first finding was that overall coma patients have a steeper 1/f slope in the 1 to 20 Hz range compared to healthy controls, which you can see here in the logarithmic space, confirming what others have observed in sleep or anesthesia. Next, we examined the spectral exponent at a single patient or control level. What we observe is that in the 2 to 40 and 2 to 20 Hz range, the spectral exponent was steeper for survivors compared to controls, similar to LZ complexity. By contrast, for non-survivors, there was again a bimodal distribution, with some non-survivors showing a steeper exponent than controls and others showing an almost flat one. One consideration here is that patients in our cohort were sedated and comatose, which means that we cannot exclude the possibility that some of these findings may be driven by sedation. However, sedation levels were similar both for survivors and non-survivors, which suggests that the differences in 1/f and also complexity in the two patient cohorts are rather due to differences in the integrity of brain circuits between survivors and non-survivors rather than sedation. Last, as previous studies have made links between the spectral exponent and the LZ complexity, we assess their co-occurrence. We observe a remarkably high correlation between LZ complexity and power spectral exponent, both for patients and for controls, which suggests that both of these measures capture similar properties of how structured or noisy are EEG responses to external stimuli in time. To summarize, in this talk, we examine different facets that auditory EEG responses can reveal about pathological loss of consciousness. First, auditory deviance detection mechanisms can be preserved in acute coma in the absence of consciousness. Their progression from first to second day of coma is informative of the patient's chances of regaining consciousness. Second, we examined the electrophysiological properties, and namely synchrony and complexity of auditory EEG responses in the first day of coma. We observed that the phase locking of EEG responses to sounds is a predictor of patients' outcome, so that survivors have a higher phase locking than non-survivors. By contrast, complexity or the spectral exponent were not informative of patients' outcome. If we consider the broader context of studies investigating the neural correlates of consciousness, one main consideration is that here we focused on the first day after coma onset, which is a window in pathological loss of consciousness that is relatively under-investigated. Future research can examine how these findings translate to more chronic states of pathological loss of consciousness. Overall, what our results suggest is that in the acute coma phase, there is a complementary role for synchrony and complexity with respect to conscious processing. On the one hand, neural synchrony differentiates patients' outcome and is a prerequisite for the presence or return of consciousness. On the other hand, neural complexity needs to find a balance between high or low values to support conscious processing. With that, I would like to thank my group at the University of Bern, and in particular Sigurd Alnes, who did all the work for the second part of this talk. Also, I would like to thank our collaborators in these projects, especially our collaborators from the Lausanne University Hospital, where these data were recorded. And last, I thank you all for your attention and look forward to any questions that you may have.
My name is Marzia de Lucia from the University of Lausanne in Switzerland, and today I would like to give you an overview about my research on the role of heartbeat signal in auditory regularity processing in conscious and unconscious states. Neural monitoring of heartbeat signals is essential for survival. In addition, as these signals are widely available across multiple cortical sites, recent research has started to investigate their role in shaping consciousness, including perception and sensory processing. Here, we are interested to understand whether the human brain can take advantage of heartbeat for improving auditory regularity processing in a variety of conscious and unconscious states. We devised a novel experimental design where sounds could either occur in synchrony or asynchrony with the ongoing heartbeat as recorded by the ECG signal. In the synchronous condition, we detect online the R peak of the ECG, and we scheduled sounds in a way that the R peak to sound onset is kept fixed along the experiment. In the asynchronous block, sounds presentation is also locked to the R peak of the ECG, but this time the ECG is recorded on a separate baseline block, which results in a natural variability between the R peak of the asynchronous regularity and the sound onset. In both the synchronous and the asynchronous condition, we omit 20% of the time randomly the sounds, and we hypothesize that only in the synchronous case, the brain can implicitly learn the contingency between R peak and sound onset. We assume that in the synchronous condition, upon omission of this expected sound, the brain will manifest this unfulfilled prediction through a surprise effect that we can measure at the level of the EEG signal. In the asynchronous case, because there is no contingency to learn because of the variability between R peak and sound, this surprise effect should be highly suppressed. In order to verify this hypothesis, we record the EEG signal in a cohort of healthy participants and concomitant ECG signal. In the experiments that I will show you in the following, I will also show you results from a cohort of comatose patients and in a group of healthy participants during sleep. The first comparison that we considered is the omission response locked to the R peak in the synchronous versus asynchronous condition. As you can see, this evoked response is highly contaminated by the ECG artifact, which nevertheless is present in both conditions. So we assume that the comparison between these two evoked response is only modulated by the synchronicity difference between these two blocks. Cluster permutation statistical test revealed a sustained difference starting at 150 milliseconds post-stimulus onset. This difference cannot be trivially due to some carryover effect of the neural response to sounds which differ between synchronous and asynchronous condition, because we found similar results also when comparing the omission response in the synchronous versus baseline. No such omission response was present in the asynchronous block versus baseline. So overall, we interpret these results as a consequence of the unfulfilled prediction in the synchronous condition upon omission of the expected sound locked to the R peak. It is possible that there is a surprise effect in the asynchronous condition, but this is not locked to the R peak. So we checked whether locking the trial by trial response to the average sound-to-sound interval for each single participant in the synchronous case, this would result in a surprise effect. The absence of such a difference between the asynchronous and baseline suggests that the jitter between sound to sound in the asynchronous condition is sufficient to suppress overall the surprise effect. Control analysis show that this differential EEG activity is not trivially due to some other parameters that differ between synchronous and asynchronous case. For example, there was no difference in the ECG, no difference in the interbeat or sound-to-sound interval. The only significant results that we obtained were due to the synchronicity that we imposed in the synchronous condition, resulting in a variable R to sound onset and more variable sound to R onset in the synchronous versus asynchronous condition. Healthy participants remained largely unaware of this experimental manipulation. In order to quantify whether there was any accidental awareness of the synchronicity between sound and heartbeat, we recorded separately the ECG in a group of 20 participants. We explained what kind of experimental manipulation we were considering, and we consider an extra block where sound-to-sound interval this time was kept fixed and matched to the interstimulus at the single subject level. We asked to rate two features in the sequence. First of all, the subject had to rate the synchronicity between heartbeat and sounds at the level at the end of each block, and also they had to rate the level of regularity in sound-to-sound onset. At group level, we observed no difference in the cardio-audio synchronicity rate across the different regularity types, while subjects could successfully rate the regularity of the fixed condition higher than the synchronous and asynchronous case. So overall, we interpret these results thinking that the EEG results of the surprise effect occur largely implicitly. And this made us wonder whether this surprise could be elicited by the cardio-audio coupling even in a fully unconscious condition. In order to verify this hypothesis, we collected EEG data with the same experiment in a cohort of comatose patients during the first day after coma onset. These patients are in coma following a cardiac arrest, and during the first day of coma, they are sedated and kept at low body temperature. All this condition results in a deep unconscious state. At 3 months, we collect their outcome, and we divide the cohort in patients that would survive and patients that would die at 3 months. In patients with good outcome, 17 of them, we found that the auditory omission response in the synchronous and asynchronous case is modulated by the synchronicity and at later latency than in the healthy condition. When comparing the omission response to baseline, we found that only in the synchronous regularity, there was a differential effect starting at similar latencies than those observed in the synchronous versus asynchronous case. So overall, as in the healthy participant, we interpret these results as saying that even in the unconscious state, the human brain can track the synchronicity between heartbeat and sound, and upon omission of the expected sound, the human brain elicits this surprise effect at latencies that start at about 230 milliseconds post-stimulus onset. Control analysis excluded that these responses were trivially driven by some other parameters such as the ECG artifact, the heart rate, or the interstimulus variability. In the comatose patients with bad outcome, instead we found no such a surprise effect. There was no differential response in any of the comparison considered, which we interpret as a possible consequence of the degeneration of the neural circuitry, which subtend the auditory processing and possible prediction in auditory irregularities. Next, we investigated whether the heartbeat itself reacts to a sudden omission in auditory irregularities. And we did so in a separate group of healthy participants during wakefulness, in the same participants during sleep across the different stages, and in the same group of comatose patients. We measured the R to R interval before the omission, during the omission, and just after that. Results showed in the group of 26 healthy participants that upon omission of the expected sound, the heartbeat decelerates, and such deceleration lasts for at least 3 trials, only in the synchronous condition. It is as if the brain teaches the heart to wait for the sound when the sound does not occur, after having learned that there is always a sound after the R peak of the heartbeat signal. This effect was highly significant, and post-hoc analysis showed that this is largely due to the synchronicity deceleration. In healthy participant, during N3, which is the deepest stage of sleep, we found a similar deceleration. In N2, similar results, and N1 as well. The only stage where this deceleration was less striking was during REM, where results were still significant, but with a less conservative threshold, and again, only in the synchronous condition. Comatose patients showed deceleration upon omission of the expected sound, similar to the healthy controls. Comatose patients with bad outcome failed to exhibit such a modulation, instead paralleling the EEG results that showed no significant effects. Overall, these results show that the brain can implicitly monitor the regularity that is established across heartbeat and sound, and that this monitoring subserves prediction of future sensory events. This sensory prediction can occur in conscious and unconscious states, and is paralleled also by this cardiac modulation following unexpected omission. Overall, it is possible that the human brain capitalizes on bodily signals beyond the heartbeat in order to generate prediction about sensory environment. Future research will help to figure out how the human brain does so across different types of bodily signals. And the cardiac response to unexpected omission represents one very easy marker that one can measure in a different type of unconscious states. In comatose patients' outcome, it is possible that this can become one marker for predicting their prognosis, as it is modulated by whether patients will survive or not. And with this, I would like to thank my team, my collaborators, and you for your kind attention.